We tend to think of evolution as this grand, branching tree of life, with species becoming more and more different over time. But what if two completely separate branches, say a tobacco plant and a soybean plant, independently evolved almost the exact same solution to a common problem? It turns out this is happening all the time in the microscopic war between plants and pathogens, and it's called convergent evolution. This idea is completely changing how scientists think about engineering disease-resistant crops.
It's a fascinating concept. You can think of it like two different engineers who have never met and used totally different materials, somehow designing the exact same key for the same lock. In this case, the lock is a molecule from a pathogen, and the key is the plant's immune receptor. Finding these convergent keys is a huge deal because our old methods of just looking for genetically similar keys across different plants often came up empty.
Right. So instead of getting stuck on the genetic differences between species, this convergent evolution concept lets us hunt for functional similarity. It suggests we could potentially borrow an immune response from one plant and give it to another, even if they aren't close relatives. It's a pretty radical idea.
Exactly. It opens the door to a kind of mix and match approach to plant immunity that wasn't really on the table before.
Fascinating. So, with this concept of convergent evolution in mind, the researchers decided to put it to the test by focusing on a specific microbial molecule and seeing if they could find its receptors in different plants. Let's dive into how they first identified these receptors in a model plant.
Right. The first step is always to establish a clear baseline.
The researchers first focused on the model plant times, Nicotiana benthamiana *, which is a relative of tobacco, to find its specific receptor for a molecule from a pathogen called PSRLK6. Using advanced gene editing techniques, they identified two proteins, NBRKR1 and NBRKR2, which work together to perceive this threat, with NBRKR1 being the primary detector.
And this is a crucial step. By identifying the exact players in a well-understood model system like tobacco, they created a benchmark. It's like having a confirmed key in their hands. Now they have a system where they can test other potential keys from different plants to see if they fit the same lock.
Right, so they've established the baseline in tobacco. The next big question then is whether they can find a similar yet genetically distinct receptor in a key crop plant like soybean. How did they tackle that challenge?
Well, this is where things get really futuristic. They brought in artificial intelligence.
To find the soybeans version of the PSRLK6 receptor, the team developed a novel pipeline combining AI structural prediction with a tool called AlphaFold 3 and then ran functional tests. This sophisticated approach successfully identified a protein called GMRLP30 in soybean as the receptor for PSRLK6, even though it shares less than 50% genetic sequence identity with the tobacco receptor.
This is the absolute core of the breakthrough. Sifting through a plant's entire genome for the right protein is like finding a needle in a haystack. Using AI to predict the 3D shape and how it might physically interact with the pathogen molecule narrows that haystack down to a handful of needles. And the fact that the soybean receptor works perfectly despite being so genetically different is the smoking gun for convergent evolution in action.
So the AI didn't just find a times similar times protein, it helped pinpoint one that times functions times identically for this specific task, despite being from a completely different blueprint. This suggests we're not just looking for close relatives anymore. We're hunting for functional doppelgangers that nature has independently created.
And that directly solves the problem you mentioned at the start. So for decades, we've been stuck trying to find these genes in crops by looking for their genetic cousins, and it often failed. Now we can use AI to find these functional equivalents, which is a much more powerful strategy.
Absolutely. So they found the receptors in tobacco and soybean. But what happens when a plant has times lost times this ability? The researchers investigated this in pepper plants.
Yes, and this part of the study is just as remarkable.
In pepper plants, the researchers found that the genes responsible for recognizing that same PSRLK6 molecule have degenerated. One gene is missing entirely and the other one is broken. This means pepper plants are effectively blind to this specific threat.
That's a great way to put it. Nature effectively turned off this particular immune switch in pepper over evolutionary time. But the truly amazing part is what the researchers did next. They showed they could turn it back on By taking the functioning receptor genes from either tobacco or soybean and inserting them into the pepper plant, they completely restored its ability to detect the pathogen.
It's a powerful demonstration of how we can engineer resistance. This leads us to the broader implications and the overall pipeline they've developed for finding and engineering these crucial immune receptors.
It really brings the whole story together into a practical toolkit.
The study proposes a novel, two-tiered integrative pipeline for identifying these convergent receptors in crops. It combines that rapid identification in model plants like they did with tobacco, with AI-guided prediction and validation to find the equivalent in major crops. It basically bridges functional genomics, AI, and genome editing.
And that pipeline is the real-world takeaway here. It's not just a one-off discovery, it's a repeatable, streamlined process. It dramatically cuts down the time it takes to go from a mysterious plant disease to having a specific gene in your hands that you can use to confer resistance. This is what will accelerate the development of hardier crops.
So, to wrap up, David, if our listeners are going to take away just a few key points from this incredible research, what should they be?
I think there are four big ones. First, the idea of convergent evolution in plant immunity is real and powerful. Different plants are inventing the same solutions independently. Second, the researchers proved it by finding the specific receptor in tobacco and then using AI to find a functionally identical but genetically different one in soybean.
Got it. And the third point?
The third is that amazing experiment with the pepper plant. It proves we can synthetically fix a broken immune system in one plant using spare parts from a completely different species. This isn't theoretical. They did it, and it enhanced the plant's resistance to infection.
And the final takeaway?
The final point is that this all combines into a new, powerful pipeline. By blending lab work and model plants with the predictive power of AI like AlphaFold 3, we now have a much faster road to discovering these crucial immune genes and engineering the next generation of disease-resistant crops. It's a huge step forward.